I'm being very, very careful, very steady, right? This is the broth tubes, and the growth characteristics in a particular broth tube, if you shake it up, you lose all of that information. So I'm being very, very gentle. All right, and again, since I'm handling this, I like to handle this without my gloves. So once I take this off, I'll go ahead and put my gloves on, because then I'm going to be handling the culture bacteria. And now remember, the plates that we had from last exercise had a lot of bacteria in that. And that's what we're very careful about, so that we don't put that anywhere other than where it belongs in the bleach bucket, right? And so these tubes, now that they've been growing for 48 hours, and if you first look at results and there's not much, you can wait another 24 for 72 hours, okay? They have a lot of microbes in them, so definitely be very careful as always, okay? All right. All right. Can't get enough of that bleach, right? Okay. And so then, of course, this will go into my fresh bleach bucket, kill everything that may have been there. And now I just bleached that, but I didn't bleach that to kill microbes that I've been working with, because I did that at the end of last exercise. This is more to disinfect the surface that I'm about to work on. So microbiologists always disinfect the surface before they begin, as well as when they're done, okay? And so I'm going to be very, very careful about not disturbing these. So what we have here is the control. This is an uninoculated nutrient broth. So we did not use this last lab. I just go ahead and added this to the tray as a control. And so what we can see is that this one we compare. So if we compare it to all of these and we put this pen behind it, hopefully you'll see that the one that I'm holding, you can see that pen very, very easily. And the other ones, you can still see it easily, but there's a haze. There is turbidity, right? And so if we go ahead and use this control, we can also see at the bottom, there's essentially nothing there, right, except optical disturbances. Here, look at that big plug of microbes next to it. So that is a sediment, and that's flocculant, okay? So this is both turbid. The one that I'm holding right here is both turbid and flocculant, and the flocculant is sediment because it's at the bottom. If we compare these two, right, then what we have is essentially there is still sediment at the bottom, okay, but in that particular instance, it is not flocculant because it is just all on the bottom. It's like grains of sand, okay? And now if we look close to this next one, hopefully what you can see where I'm pointing is that there's a lot of material at the top of the tube. And if you look kind of around the whole thing, what you'll see is that there's essentially like that right there, that area, that goopy stuff all around it, that is hydrophobic material stuck to the side of the tube. So when we look at this, what we see is that the tube going up has a little bit of turbidity, but you can see through it. When you get to the top, what we see is that essentially it's this big snotty material that is like a plug at the top. Now when I swirl it around, it will be disrupted, but this is called a pellicle. So if you have some like goopy stuff at the top that's kind of growing up the side, call that a pellicle. Now that's different maybe than over here, because this right here, that is just an optical illusion from the tube, I think, whereas here you can actually see the material. So that is an example of a pellicle. And then zoomed out, you can see that this is still the one with the pellicle. Okay, and then we go to this one, and essentially there's no sediment, but there is turbidity. And you may not be able to see it, but the very top, there are no microbes at the very top. So the turbidity ends around here, which means that they are not, well, I'll let you do the reading and tell me what kind that is, but they're not at the top. If you're ever wondering if something has a sediment, okay, so for example, if I look at this one, it's got a sediment, what you can do, make sure it's capped, you can go ahead and swirl it around, and sometimes what you'll see is, can't see it too well, but stuff is starting to come up and float up to the top. And you only want to do that after you've done everything. So before you start moving it around, the first thing you should do is you should take a photo, just hold it up like this, right? Put a nice background behind it, whatever the case may be. And that's up for you to decide, but put a nice background behind it so that you can see everything, okay? And then take a photo of it. So for the top, you can take a photo of them when they're in this particular rack. But the problem with taking a photo of only in the rack is essentially that you don't see the bottom, right? So you could take a photo like this, or you could go ahead and just also hold it up like this, but we need to see the whole tube. So if I were to hold this like this and then Greg were to come in and take a photo of it, then in Photoshop or PowerPoint or Word, I could go ahead and put the labels on it. Remember, for your photos, I need to see what is here in particular. And in general, everything needs to be labeled very clear. Remember to crop. So once you first open this up, take nice pictures of them, analyze them, enter the data, right? Have your assistant there, and your assistant can write down your notes for you, right? Have somebody share in the delight of this microbiology, okay? So what we're going to do next is we're going to go ahead and take microbes from the broth, and we're going to streak them onto plate for the next exercise in this, which I like calling from broth to plate.